afternoon, everyone. So this will serve as your video lesson pertaining or continuing our topic in microbiology, and just to continue the microorganism or types of microorganism that we've already discussed, let's proceed to another very important microorganism. And aside from bacteria, this is also another microorganism, sorry, and that would be virus. Okay, so this chapter is pertaining to virology, or the study of viruses. So our topic, our main goal for today is to know the structure, classification, and multiplication of viruses. Not only that, we will be knowing also how to cultivate and replicate viruses, a non-viral infection agent, and of course a teratogenic or oncogenic process of a viruses. So as you can see in our references, you can view a microscopic view in virus. So this is one of good example or picture of viruses in action pertaining on viewing it in a microscope. So another one is the human rhinovirus or the common cold. That is how you viewed, or that illustration or that picture can be seen in microscopic diagram viewing. Okay, so as we all know, viruses have a host range. It is infect specific cells or tissues of specific host or specific bacteria of specific plants, and meaning to say viruses itself is, so again, meaning to say, when we say virus or viruses, these are microscopic organisms that can infect a host. So that's why it's said in our references that viruses have a host range, or different host to be injected, and we can literally explain that the host race of viruses is on specific, if it is cell, tissue, or even the host of bacteria, or even in plants. So not only that, when we say host range, meaning to say viruses can be in humans, plants, or animals. There are actually a small piece of genetic information from our genetic material, from DNA or RNA, and it is located inside of a protective shell called capsid. So we've already done discussing the parts of the cell itself. So some viruses also have an envelope. Viruses, and always remember that viruses can't produce without a host. Some common diseases caused by viruses included flu or the common cold, or even the COVID-19 is one good example of viruses. So aside from that, it's also said that viral specificity refers to the specific kinds of cell a virus can infect. It is regulated by the specificities of attachment, penetration, and replication of the viruses. Meaning to say, when the viruses attach to the host, it also penetrate and replicated. Okay, that's why it's not only, this topic is not only focusing on the structure or even the classification of the virus, and it is also focused on the multiplication and replication of the viruses. So that explains the definition of viruses. So again, when we say viruses, it has or have a host range, or a huge of host range, meaning to say viruses can attach into a specific cell or even tissue, or even a specific bacteria, or even a plants. Why? Because as we all know, viruses or the microscopic organism that can infect host. There are usually a small piece of genetic information that is located inside the protective shell of our DNA or an RNA, which is called capsid. And some of the viruses also have its own envelope, and it can reproduce without, or it cannot reproduce without a host. It is different with regards if we will be comparing into bacteria, because bacteria can reproduce without a host, because as we all know, bacteria has its own single cell, or it is a unicellular organism, while viruses, it cannot reproduce without host. So that is very good explanation that viruses will live only if there is a host. Okay, so some common diseases or example of bacteria is caused by viruses, and that is included into flu or common cold. Usually viruses that can usually that viruses, that a very good example of viruses is a COVID-19, which we've already encountered in the year of 2019 onwards, right? So that is a definition of viruses. So now let's move forward on the properties of virus. So viruses are not cells. So that's actually correct. When we say not cells, it cannot live without the host because it no living cell. It has no living cell. So that's why it's very we can compare the viruses to bacteria. Why bacteria can live without a host because bacteria has a single cell, while viruses have no cells. And it has since there is no cell, of course, this the command center of the cell. Viruses has no or do not have a nuclear or mitochondria or even ribosome or other components of the cell. Kasi number one, again, viruses are not cells, or this kind of microorganism has no cells. Viruses also replicate and multiply. Tama. It dumadami, dumodoble, tumitriple. It replicates by its own virus or by its own itself, and it multiply. Viruses do not grow. Okay, but it multiply or replicated with the same identity of that viruses, but do not grow. But always remember that viruses do not grow. Viruses replicate or multiply only within the living cell. So it replicated with with the same identity of virus in when the viruses is in the living host. Okay, viruses are obligate or intracellular parasite, which means by the going back to the definition itself, it contain either an RNA or the ribonucleic acid or DNA or the oxyribonucleic acid genome surrounded by a protective virus-coded protein coat. So that's why it's called a obligate intracellular parasite. Why? Because going back to the definition itself, viruses can contain either RNA and DNA. And not only that, viruses may be viewed as a mobile genetic elements, most probably of cellular origin and characterized by a long coevolution of virus and host. That's why viruses can explain into huge range of host. Okay, the term virus was coined by, of course, the man itself, Louis Pasteur, and it is from the Latin word which is called poison. Okay, so what are those components of viruses? So these are, number one is a virion. Virion is an infectious virus particle. Not all viruses actually has its own infectious. Not all viruses are infectious, okay? Or not all virus particles are infectious, okay? But if we literally define infectious virus particles, that is called virion, okay? Viruses are composed of nucleic acid like RNA or DNA or never both. It can compose a nucleic acid. Why? Because as we all know, nucleic acid is a good component of RNA and DNA. And going back to the definition of viruses contain either RNA or DNA. But please coin with this. Please remember this. Viruses can also never both, or can also not contain either RNA or DNA genome. Okay? So all the viruses have protein coat, of course. That's why it's called intracellular parasites, because it can be viewed in the mobile genetic elements or most of probably a cellular origin, and it can characterize in a coevolution of virus and host. Okay? And that surrounds and protects the nucleic acid core. Okay? Some viruses have a lipid envelope or membrane. This is what we define that viruses has its own envelope, and that envelope is lipid envelope, and that is particular into fats or membrane surrounding a nucleocapsid core. The source of the envelope is from the membrane of the host cell, and some of the viruses package enzymes is particularly on the RNA-dependent polymerase or the other enzymes. Some do not package enzymes. Okay? So other of it, or this some virus packages enzymes is just an example of a viruses. So it can located in the dependent RNA polymerase or other enzymes, or some of the viruses has no or do not have a package of enzymes. Okay? So not only that, if we literally know the components of viruses, we will be moving on the size and comparison of viruses, how big they are. So, as you can see in the references, you can definitely know the size of the viruses. So I will not be reading it, just familiarize the sizes of the types of viruses. We also have this epoxyvirus. It approximately 250 nanometer, and herpes simplex, it is 150 nanometer. So it's just a comparison how big the viruses to the bacteria cell. Okay? Not only that, of course, if we're talking about viruses, so now let's proceed to the other main topic or main goal for this discussion, and that would be the size and morphology of the viruses, and also the protection of the virus, which is capsid, the other envelope, the complex, and the nucleic acid. So let's start first on discussing the two major structure of viruses. So as we all know that there are two major structure of viruses, it is called naked nucleocapsid virus and the envelope virus. Okay, so as you can see, this is the differences between the naked nucleocapsid virus and the envelope virus. So from the description itself, you can literally see the difference between the two, because the envelope has its own another protection, so another outside layer, while the naked, it's just a there's no another protection or another layer of envelope. So that is the differences between the two, and we have also the parts of it. So I will not be reading it already because it's already been seen in our references. So now let's proceed on knowing what is capsid. Capsid is very important in the structure of the virus. Why? Because it is a protective outer shell that surrounds viral nucleic acid. So the same with bacteria, capsule is, or capsid or capsule is this somewhat the same, because it is a protective outer shell that surrounds the area of the structure of its structure. And in the capsid, there is use for binding or cell surface protein, and that is called capsid spikes. And if we're trying to know the subunits or collectively protect, or these are the subunits that protect the nucleic acid from the environment, that is called capsomer. So that is how to identify what is capsid. And not only that, of course, the major complex or mechanical properties or the main function of the capsid is to encapsulate the viral genome in one host. So meaning to say, it transport and subsequently release it inside another, or in the inside of another host cell. So that is the main function of the capsid. So since we literally identify what is capsid, let's move forward now to identify what is envelope. So surrounds the envelope is called a lipid protein. This lipid protein basically is a modified version of our cell membrane. As we all know, cell membrane itself in parts of the cell is composed of fats or lipids. It composed of hydrophobic and hydrophilic, or the water-faring and the water-loving. Not only that, if the capsid spikes has its own binding cell surface protein, envelope has its own binding cell surface protein, and that is called envelope spikes. During a release of animal viruses, a part of the host membrane is taken, and that would be the envelope, or the part called envelope. And not only that, the main function of the envelope viruses is to contain a viral genome, and the core proteins wrap within one or more membrane. So that is literally how to identify what is envelope. So now let's move forward to another term or part of the structure of the viruses, and that is nucleic acid. So literally, nucleic viruses contain either DNA and RNA. So supposedly, when we say it is contain DNA and RNA, so meaning nucleic acid is there, or nucleic acid occurs in viruses. It possesses only the genes to invade and regulate the metabolic activity of the host. And one good example of it is hepatitis B and herpes viruses. So no viral metabolic genes or the viruses uses the host metabolic resources. So nucleic acid is basically occur in the viruses because viruses itself has contained either DNA or RNA. But remember, sometimes viruses cannot contain or not contain or not even has a DNA or RNA. So not literally all the time that viruses has its own DNA or RNA. Okay? Remember that one. So if you have questions and clarifications, you can ask me on our GC. Okay? So another part that is related to the structure of the viruses is bacteriophages or bacteriophages. What is this? This is actually a polyhedral head. Usually it's described as a polyhedral head and helical tail and a fibers for attachment. These are considered either lytic or temperate. These are often associated with virulence genes in bacteria. One good example of it is diphtheria toxin or Clostridium and diphtheria. Okay? So that was the bacteriophage. And not only that, when we are trying to define what is bacteriophages, it is actually also known as phages itself. It is phages of viruses that infect and replicate only in the bacterial cells. So it's a part of viruses that's why it's called bacteriophages, because it infect and replicated or even multiplied in the bacterial cell itself. So that is bacteriophages. And it is actually known for slowly kill and selectively target bacteria. They are the most common biological entities in nature and have been shown to effectively fight and destroy multi-drug resistant. And that is bacteriophages. Okay? It's formally known as phages or bacteriophages, and these are viruses that slowly kill, selectively target bacteria. Okay? So this is actually a description of bacteriophages. So from the graph itself, as you can see here from the graph, and this is one good example of bacteriophages or the T-even bacteriophages. It penetrates the host cell by specifically binding and injecting their DNA into host. So literally, it's just an example. So after replication, bacteriophages release lysozyme or that makes weaken or destroy or even rupture the cell and release numerous virions. Okay. Literally, bacteriophages can trigger and weaken or destroy the cell itself, and that makes the release of numerous virions. Okay? And of course, here we have illustration how does the assemble of the new viruses. So we will not be specifically identifying it as long as you know the flow of it. Okay, from the maturation to the lysis of weakened cell, and of course, it releases of viruses. Then afterwards, it's adsorb, then penetrate, then duplicate the pages of components. That's why a while ago I'm saying that after the bacteriophages binding and injecting to their DNA, now the bacteriophages release a lysozyme, and that makes weakens or destroy or even rupture our cell, and that can release virions. So it's like a cycle. So here pinapakita lang kung paano bacteriophages can infect or can affect or kill the target bacteria. Okay? So that makes the reference, or that explains the reference or the bacteriophages. So let's move forward to another illustration. So this is another way on explaining the structure of the viruses. So aside from bacteriophages, we also have temperate phages. So temperate phages can cause diseases. For example, Corynebacterium, diphtheria, and Clostridium botulinum contain prophages that have genes which encode for toxins. That's why, going back to the definition of viruses, that's why according to Louis Pasteur, viruses is coined from the name itself, poison. Okay, because it produces toxin. Without this propages, they do not produce the toxin. Without the toxin, no diseases. So if there is this called part propages, or these propages have a gene and it encoded toxin. But if there's no propages at all, no, with no toxin and no diseases at all, that's thus they are example of bacteria and viruses interacting to cause medically relevant disease. So this is the temperate pages formation. So as you can see, from the prophage itself, it splices the virus genome, or from the DNA itself splits, and it created the splice of viral genome. So literally meaning the description describe the integrated of prophage into their own genome, and it is called now a lysogen. This is able to transmit to its progeny the ability of produce another phage. So that is literally how that the formation of prophage and lysogeny occur. So again, how can we describe prophage? Literally, when the lysogenic bacteria carry the phage genome, and that is called prophage. It integrated into their own genome or its own prophage, and that will call or result with lysogen. And that lysogen will able to transmit to its progeny the ability to produce another phage. So that makes the temperate phage, or the formation of prophage and lysogeny. So now let's go with the classification of viruses, and it explains the structure, chemical composition, genetic makeup, host relationship, type of disease, and Baltimore classification of viruses. So this explains the Baltimore classification of animal viruses. Okay, so this another classification of viruses, that is Baltimore classification of animal viruses. So that is very important to know what is the Baltimore classification for us to know what is the subdivision of its viruses. So Baltimore classification animal viruses are subdivided into 7 groups. These are DNA viruses, which is group 1 and 2. We have also the RNA viruses, which is group 3, 4, and 5. And we also have the RT viruses, and that is group 6, and 7. So if we're trying to know the abbreviation, or the abbreviation of RT viruses, it is ribosomal transport viruses. So it is part of group 6 and 7. So the schematic diagram of it is how the viruses in each group differently synthesize their messenger RNA. So Baltimore classification will be starting on how it transport or synthesize in the messenger RNA. So meaning, after all the distinct mRNA transcription strategies representing with the different or each virus group, Group 1 is represented by virus containing the double-stranded DNA genome, while the N, it makes synthesize in mRNA by transcription from the DNA genome template. So now the group 2 will represent or will occur. It represented by the virus containing a single-stranded DNA genome, and that will first convert their single-stranded DNA genome to double-stranded DNA, which is then used as a template for mRNA transcription, and that makes the occurrence of group 3. And that group 3 is represented by viruses containing a double-stranded RNA genome, and it synthesizes mRNA by transcription from their double-stranded RNA template. And here comes now the group 4, which represented by the viruses containing a positive-stranded RNA genome, and it utilizes the genomic RNA directly as an mRNA. Take note, it is usually occur as a dotted lines, And after that, the occurrence of the group 5 will represented by the viruses containing a negative stranded RNA genome, and the group 5 viruses will synthesize in the mRNA by transcription from their RNA genome template. And now, from the last group of the Baltimore classification of animal, the reoccurrence of the group 6 and 7. This is a reverse transcribing of RT viruses. When we say RT, it is a ribosomal transport viruses, and they have either RNA or double-stranded DNA genome. These ribosomal transport viruses are not classified as either RNA or DNA viruses. It is very important feature that is shared by the RT viruses, and the viral DNAs are synthesized via transcription. Note that although group 6 viruses contain RNA in genome, the genomic RNA does not serve as an mRNA, unlike those of group 4. Okay, so that is the overall classification, or how we enables to classify all animal viruses to extend that genome replication strategy is precisely predictable. So that explains the whole illustration in Baltimore classification of animal viruses. So always remember that it's subdivided into different classes. Okay, so that's it. Aside from that, we will be also knowing the general steps. The general steps in viral multiplication. Okay, so these are the adsorption, penetration, uncoating, synthesis, assembly, and release. So from the figure 6.12 and figure 6.13, it shows the viruses recognize specific receptors, and then the viruses penetrates the cell in figure 6.13. So how does the general steps in viral multiplication do, or how does the process itself? So literally, the first step of the virus life cycle, it can be divided into the adsorption, penetration, and uncoating, or even in synthesis, assembly, and release. So literally, it started with the viral entry. So the first step of virus infections involve a recognition of viral receptor by a virus particle. Okay, so this viral entry can be divided into 4 steps, and that would be the attachment, penetration, cytoplasmic trafficking, and uncoating. So let's discuss first what is attachment, or how does the attachment occur. So the attachment refers to the first encounter of the virus particles with the host itself, which involve 2 kinds of host proteins, and that is plasma membrane attaches, or plasma membrane attachment factors, and the viral receptors. The attachment itself, or the attachment factor on the cell surface, recruits and holds the virus particles, and thereby facilitating the interaction of the viral particle with the entry receptor. So that how the attachment occur. And from the different binding of the virus particles, it promotes penetration of the virus, and that makes another way of viral multiplication, and that makes the penetration. Okay, and as I've said, that makes the next step in multiplication, or viral multiplication, which is penetration into the cytoplasm. So what happened in this penetration? So the mechanism for the penetration differs whether the envelope, or whether there is an envelope or not. So for the envelope viruses, one of the following 2 mechanism is used. Listen: direct fusion and receptor mediated endocytosis. So that makes here we have endocytosis here. And it, for non-envelope naked viruses, the receptor mediated is the endocytosis is used for penetration. So this is another way, which is membrane fusion. So that is how you, the first one is, which is the attachment, or the adsorption, and the next step, which is penetration. That explains the illustration below. And how does the animal viruses replicated? So since we've already done discussing the adsorption and penetration, let's move forward. So the general replication scheme for an animal viruses is in this illustration. So here is the step. So as you can see, in adsorption, the virus attached to its host cell by specific binding of the spikes to cell receptors. Then the next step will be the virus engulfed into vesicle, and that makes penetration. Then thereby, the viral RNA into the cell cytoplasm is now uncoated, and that leads now to explains how the viruses replicated by protein production. It is actually under the control of viral genes, and it synthesizes the basis of the components of new viruses, which can be found in RNA molecules or even in capsomers and spikes. So after it replicated, it's now go on assembly, and the proteins are inserted into a cell membrane for the viral envelope, and that viral envelope is called nucleocapsid that form from the RNA and the capsomer. And the last one would be the release, and that makes the envelope viruses bud off of the membrane, carrying away and spikes. This complete virus or virion is ready to infect another cell. So that's how the replication scheme of animal viruses. So from the introduction that I gave to you, from penetration, from the adsorption or the attachment to the penetration. So as I've said a while ago, that in penetration we have endocytosis and membrane fission. Okay. So membrane membrane fusion is basically occur in penetration. Why? Because it literally implies how the mechanism of two membranes fuse to one another, and that is the cases when the viral nucleocapsid is directly delivered to the cytoplasm, and that makes a leaving the viral envelope behind on the plasma membrane, and it penetrates by a membrane fusion. So that was I'm saying a while ago, that it can be endocytosis and membrane fusion. So do not forget that envelope viruses can be a mechanism of membrane fusion and can be an receptor of mediated endocytosis. It differs it differs whether it's envelope or not. So meaning to say, in envelope viruses, it can be membrane fusion or endocytosis. In non-envelope naked viruses, it can be endocytosis only for use in penetration. So moving forward, this is another magnified view of viral binding of envelope viruses. So worst case of HIV, which is already been released when the replication scheme is undergoing the process. Okay? Yan yung exiting from a T cells. And what happens if it is a DNA viruses? So one good example is herpes viruses. Okay? So that is just a illustration. So let's move forward on another one. Another example, we have most simple and efficient cases, like for example RNA viruses. One good example is coronavirus or retrovirus. Okay. Another illustration that give emphasis with the replication of RNA viruses, and that is explains again what I'm saying a while ago, and that started with the adsorption, penetration, uncoating, synthesis, assembly, and release. Okay, so the replication of RNA viruses in a 6.2 insight. Note the differences between a positive sense and a negative sense, and that is retrovirus replication. It is also be able compare contrast phage and animal virus replication. Okay, another terminology to be unlocked, and that is cytophatic effects. These are the damage to the host cell due to the viral infection, and one good process that can occur in a cytophatic effects is the transformation. So here is the illustration on viral transportation of cells, or also called as cytopathic effect. Okay, and next we have the cultivation and replication. This is the in vivo methods, or the laboratory animals and embryonic bird tissues, and this is we also have the in vitro methods, which is the cell or tissue cultured. Okay, so how does it be possible, the cultivation and replication? It can be possible to the study of viruses in animals, but due to the complexity of the animals, expense of animals, and political environment related to use of animals, alternatives have been developed. So chick embryos and eggs, one good example of influenza and persistent infection of transformation or even in cancer. So this is illustration of making the influenza vaccine. Okay, so going back to the cultivation and replication itself, aside from seeing the making the influenza vaccine, let's go on to the differences between in vivo and in vitro. So when we literally say in vivo, it means research done on living organism, while in vitro means research done in the laboratory dish or test tube. So the differences between the two words is that when we say in vitro, it is literally refers to experiment that is done in the laboratory within the confines of a test tube or laboratory dish. This means tissue, cell, or other parts of organism remove and place in laboratory dish is called in vitro, while in vivo is literally refers to a medical test or experiment or even procedure that is done in a living organism such as laboratory animal or human. So that makes the differences between in vivo and in vitro. Okay, so that is the cultivation, replication, and replication. So let's move forward now to the non-cellular infectious. So that these infectious agents, and one good example of that are the prions. So prions is basically discovered by Stanley Prisoner and in 1978. According to this man, prions are proteinaceous infectious agent, meaning to say it do not have a nucleic acid because it has it has a component of a protein. Prions are considered to be causative agent of Creutzfeldt-Jakob disease, scapupoperoneal spongiform or encephalopathy and Kuro. So that is considered how can you get a prions, or this is basically a causative agent of this diseases, or in a simply way on how we can understand what is prions, it is basically a pathogenic agent that are transmissible and are able to induce abnormal folding of specific normal cellular protein. And this protein is called prion protein that are found in the most abundantly located in our brain. So the function of this in a normal protein are still not completely understood. So that is a prion, or that means prions. That is literally how to explain prions. So what do prions cause in humans? So basically, prions occur when normal prion protein found on the surface of many cell. So this abnormal accumulation of protein in our brain cause memory impairment. So that is the cause of prions in the human body. So this is usually located in our brain. Okay, so this is the protein structure of two form of prion protein. We have good and bad. So this you can literally identify if the prion protein is good or bad by its own structure. So let's move forward on oncogenic potential viruses. So these are the cancer is set of diseases known to disturb the normal functioning and properties of the cell. Tumors may be malignant or benign. Malignant tumors spread by metastasis. So literally, oncogenic potential of viruses is usually discovered by Peyton Ross, and this is a kind of filterable agent that could transport a sarcoma or a type of cancer. So that is oncogenic potential viruses. So at least 6 viruses have been found to cause human cancer, and that least is the type of it. And oncogenes are normal cellular regulator genes. When modifying these genes code for gene products that disturb the normal regulatory patterns of the cell and can result in a loss of the normal properties of the cell growth and division, resulting in cancer. So literally oncogenic potential of viruses usually can be found by filterable agent called transmit intersarcome, or it is a type of cancer. So these are the type of oncogenes. We have viral oncogenes. These are usually found in retroviruses. The V oncogenes are viral homologous of cellular oncogenes, and the V oncogenes can disturb normal regulatory properties by certain mechanism. Okay? So literally, when we say oncogenic viruses, it explains, or these are the viruses caused by a cancer. So there are a lot of oncogenesis or onco oncogenic viruses that can be found, or diseases that can be found in or can cause by a cancer. We have actually another's recognized human oncoviruses. Not only it causes a cancer, of course, we have the so-called human papillomavirus, the hepatitis B, C viruses, HBV and HCV, or human T-cell lymphotropic viruses. So, or even the Merkel cell polyomaviruses, or the MCPYV. So that is example of oncogenic. So always remember that when we say oncogenic potential viruses, it means, or it causes cancer. Okay? So, one good another illustration explains with that. We have a malignant tumor formation. So how does the tumor itself form? Okay? So that ends our discussion for this afternoon. So for malignant tumor formation, as you can see, how does the tumor form from, and how it's called malignant because of this is the process itself. So it is also produced by oncogenic potential of viruses, or human oncoviruses. Okay, always remember it came from the cause of cancer. So if you have questions, clarifications, you can leave that in our GC, or you can PM me. So that explains our topic for today, and that is virology. So in our face-to-face class, we will be having our oral recitation pertaining to your laboratory, the task that I've given to you nung nakaraang linggo pa. So, prepare in advance because I will be giving you a oral recitation. So, if there's no more anything else, you can ask me in our GC. Thank you so much. Enjoy your holiday. Bye.